Good morning. It's April 22nd, and this is your daily brief in science. Here's everything you need to know. A groundbreaking study published in Nature Neuroscience has unveiled a genetic link between autism spectrum disorder, or ASD, and myotonic dystrophy type 1, or DM1. The research focuses on tandem repeat expansions, or TREs, in the DMPK gene that may serve as a common mechanism affecting gene splicing. This connection is significant as it suggests that altered RNA from these TREs binds to a protein essential for regulating gene splicing, resulting in a depletion of this protein vital for proper brain development. The imbalance of proteins disrupts gene splicing for various genes involved in brain function, potentially explaining the social and behavioral challenges observed in DM1 patients who also have ASD. Dr. Ryan Ewan, a senior scientist at RARE-seq Kids and one of the authors of the study, highlighted that these findings could lead to new diagnostic methods and precision therapies that target the molecular pathways implicated in autism. This research contributes to our understanding of autism's biological origins, indicating that there may be specific genetic causes rather than viewing it solely as a spectrum disorder. The study comes at a time when recent CDC data shows a dramatic increase in autism rates in the U.S., with 1 in 31 children affected, raising important questions about the underlying causes of this rise. Supported by organizations such as the Azrieli Foundation and the National Institutes of Health, the study underscores its significance in genetic research. Researchers are now investigating whether the missplicing observed in DM1 also occurs in other genes linked to ASD, with the aim of developing therapies that could correct these genetic issues. In related findings, Dr. Christopher Pearson at RARE-seq Kids has identified a molecule capable of contracting TREs in Huntington's disease, suggesting potential applications for treating conditions like DM1 and ASD. DM1 itself is characterized by progressive muscle weakness and cognitive issues due to a malfunctioning gene. These findings underscore the need for targeted support and potential gene repair treatments for individuals affected by both DM1 and autism. Additionally, a non-invasive brain stimulation treatment known as transcranial pulsed current stimulation, or TPCS, has shown promise in improving certain autism symptoms in children aged 3 to 14, indicating ongoing research into effective treatments for autism. Researchers at Stanford Medicine have introduced a new blood test called RARE-seq, which significantly enhances the detection of cancer-related RNA signatures. This innovative test has shown promising results in clinical trials, identifying cancer-related RNA in 101 out of 139 lung cancer patients. Detection rates improved notably, rising from 30% in stage 1 patients to 83% in those at stage 4. RARE-seq outperforms traditional DNA-based detection methods, demonstrating its effectiveness across various stages of cancer. The method analyzes cell-free RNA in the bloodstream, capturing RNA fragments released from dead cells, including those from tumors. The study involved 437 plasma samples from 369 individuals with varying cancer stages, benign conditions, and healthy controls. One of the key advantages of RARE-seq is its ability to monitor treatment resistance resulting from changes in cellular behavior rather than just genetic mutations. This expands its diagnostic capabilities beyond cancer detection. Additionally, the test shows promise in identifying lung damage in healthy smokers and monitoring respiratory distress. The RARE-seq method includes advancements in blood collection, RNA extraction, and library preparation, which enhance detection accuracy. Researchers developed techniques to filter out RNA from platelets that could obscure cancer signals, allowing for effective analysis of both fresh and frozen blood samples. The findings were published in the journal Nature on April 16, 2025, with contributions from several prestigious institutions and support from multiple NIH grants. These preliminary results indicate that RARE-seq could have broader applications in diagnosing both cancer and non-cancer conditions, pending further clinical trials. Researchers at UC Riverside, led by Vincent Lavallo, have achieved a significant breakthrough by generating and isolating a stable carbene in water, a task once thought impossible. This accomplishment validates a 67-year-old hypothesis by Ronald Breslow, which proposed that vitamin B1 could form a carbene to facilitate crucial biochemical reactions. The findings were published in Science Advances on April 21, 2025, marking a milestone in resolving a long-standing biochemical mystery. The ability to stabilize carbenes in water carries profound implications for the pharmaceutical industry. These reactive intermediates can function as ligands in metal-based catalysts, potentially leading to cleaner and safer production processes. By utilizing water as a solvent, this research aligns with the principles of greener chemistry, reducing reliance on harmful organic solvents while taking advantage of water's abundance and non-toxic nature. This discovery also enhances our understanding of natural cellular chemistry, which predominantly occurs in aqueous environments. It allows researchers to generate and study reactive intermediates in water, which is vital for mimicking the complex chemistry that takes place within living cells. The stable carbene can be analyzed using advanced techniques like nuclear magnetic resonance spectroscopy and X-ray crystallography, providing robust evidence of its existence. Lavallo reflected on the personal significance of this achievement, considering the evolution of carbene research over the past 3 decades. Varun Raviprolu, the first author of the study, emphasized the importance of perseverance in scientific research, illustrating how seemingly impossible ideas can become reality. This discovery not only supports Breslow's hypothesis, but also serves as a motivational reminder of the transformative potential of scientific breakthroughs. Researchers at the Indian Institute of Science have announced a revolutionary battery technology that not only generates hydrogen peroxide but also cleans wastewater. This dual-purpose solution addresses both energy storage needs and industrial pollution challenges. Professor Aninda J. Bhattacharya emphasized that this innovative approach is sustainable, economically viable, and highly energy efficient, making it suitable for widespread applications. The research, published in ACS Applied Materials and Interfaces, utilizes zinc-air battery technology, which is recognized for being a low-cost and energy-dense alternative to traditional lithium-ion batteries. The process involves a reduction reaction at the battery's cathode, where oxygen is converted into hydrogen peroxide using a metal-free, chemically modified carbon catalyst. Lead researcher Asutosh Behera highlighted the significance of generating a disinfectant while simultaneously degrading pollutants through this integrated process. This work is part of a larger trend of rethinking battery chemistry to explore multifunctional energy systems that not only store electricity, but also facilitate valuable chemical reactions. By re-engineering the zinc-air battery, the team has enabled it to produce hydrogen peroxide during the discharge cycle, moving away from conventional, energy-intensive methods that rely on costly metal catalysts. Traditional hydrogen peroxide production methods, such as the anthraquinone process, are energy-intensive and dependent on fossil fuels and precious metals. The iisc approach avoids these issues by utilizing abundant zinc and ambient air. This technology is particularly promising for rural or off-grid areas, as it can provide both power and clean water, with potential applications in medical sterilization and wastewater purification. When applied to toxic synthetic dyes from the textile industry, the generated hydrogen peroxide effectively breaks down these pollutants, significantly reducing color and toxicity in wastewater. Researchers have made a significant breakthrough in the search for distant exoplanets with the development of an innovative coronagraph. This new device effectively blocks out light from bright stars, enabling astronomers to detect faint exoplanets that current telescopes struggle to resolve. Nico Deschler, the research team leader from the University of Arizona, emphasized the challenges faced in identifying these elusive exoplanets, which are often overshadowed by the brightness of their host stars. The coronagraph utilizes a spatial mode sorter to isolate the light from the star, allowing for the capture of images of off-axis exoplanets, similar to how different acoustic frequencies can be isolated from a piano. In testing, the coronagraph achieved a remarkable contrast ratio of 1,000 to 1 between a simulated star and an exoplanet, successfully estimating the position of the exoplanet. Current efforts are now focused on enhancing the mode sorter to reduce crosstalk, which can interfere with image quality at the extreme contrast levels necessary for exoplanet studies. This new coronagraph design promises to provide more detailed information about sub-diffraction exoplanets, potentially aiding in the detection of biosignatures. It could reveal Earth-like planets located in the habitable zone, which can be up to 1 billion times dimmer than their stars, making them extremely difficult to detect. The proof-of-principle experiment may also pave the way for new optical pre-processing methods in astronomical instrumentation, with potential applications extending to quantum sensing and medical imaging. This research aligns with NASA's Habitable Worlds Observatory, which is dedicated to advancing exoplanet science and inspiring various coronagraph designs with different performance trade-offs. Directly imaging exoplanets remains particularly challenging due to their proximity to bright stars and the significant brightness differences. Thus, the capabilities of this coronagraph are especially valuable, demonstrating the potential to achieve the fundamental limits of exoplanet detection and localization as defined by quantum optics, capturing images with enhanced resolution. By revealing Earth-like planets in habitable zones, this device could provide crucial insights into the potential for life beyond Earth. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.